Hello there, it's my wonderful pleasure to have you guys with me on the lecture today. And on this edition of the video, we'll be solving paper 2 biology past questions, AQA. But please bear in mind that these questions, or let me say the topic genetics in this is not it's not only peculiar to AQA. Whether you're doing Excel and all of that is still relevant to you. All right. So the way it's done is, okay, try to attempt the questions first, and afterwards see if you get the answer. You just pause the video to answer the question, then afterwards you play to see if you have the answer or not. So question 1 says hemochromatosis is an inherited condition that causes an accumulation of excess iron in the body. So hemochromatosis is caused by a recessive allele. So what is an allele? Well, an allele is the alternative form of a gene. Now, this allele could be homozygous. So if usually we use letters to represent alleles in genetics. So if they are the same case, uppercase, or they are both lower cases, that's homozygous. All right. But if it is one bigger case, uppercase, and one lower case, or say something like these are heterozygous. All right. You might want to check up my lecture on that. But really, I just decided to add, if you just write that definition is enough, but I added that for emphasis sake since it's a revision. Now, when does an allele control development of a characteristics? Then hear this. Alleles could be dominant when it is represented in uppercase, or recessive when it is represented in lowercase. But whenever you have alleles in this way, this is homozygous, heterozygous, I mean to say, heterozygous representation of the two alleles. Why is it heterozygous? One dominant, one recessive. Now, whatever this is representing will be what will be seen in the organism. So that means that if this was representing black and this was representing white, that means this organism will be black. Why? Because this is a dominant allele. It's going to dominate over the white. However, if you have recessive allele, please, in homozygous form, then it's going to dominate. What do I mean? We said earlier, I wrote A and A. This is dominant, this is recessive. So whatever this is carrying is what's going to show the organism. But when you have something like this, then we said this was black and this is white. So apparently, if you have white, then this guy is going to be white in color. So to cut the story short, all of this explanation was to make you understand. So when does recessive allele control a characteristics? So the answer will be when it is in homozygous form. All right. Now, a woman with the hemochromatosis wants to have a child with a heterozygous, a man, please. So the man is heterozygous. A man. Okay. 1 second. Now, the reason why I did that was this, please. I came back here to check something. It says it's caused by a recessive allele. That means that for you to have this disease, it must be in homozygous form. So let's assume from here that the H is used to depict that. So when you say a man with, that means he has it. So his gene, the two alleles in his genes will be homozygous form. Got married to a woman who is heterozygous, all right? So that woman is going to be one big H and one small h. So it says here, figure 1, complete the figure to determine the probability of the children having hemochromatosis. So circle any child who would have chromatosis. So that means any child that has this will be having the disease. Now, use the following symbols to represent the allele. So, dominant allele and recessive allele. So, the man is, these are his genes. So, these are his alleles. The woman, these are her alleles. All right. So, she's heterozygous and the man is homozygous. The reason why we said, because you wonder why did I just pick, how did I know he's homozygous? Now, because it says here, a man with hemochromatosis. And it's being said at the beginning here that for you to have hemochromatosis, it is controlled by a recessive allele. And we concluded that any trait controlled by a recessive allele will only work or show in an organism phenotypically, physically, when it is in homozygous form. All right. So that's why we knew, or we know, that this is going to be homozygous. So if you cross that, I believe you know what this is. This is called Punnett square. So HH, then this goes down, this comes here. So it's going to be HH. All right. I'm doing that because of those that might not know. So this is going to be H, big H, small h, then small h, small h. So now it says here, circle any children who would have hemochromatosis. Then hemochromatosis can only be present when you have this allele present. So what you're meant to do is you're meant to look at this table and see where do we have such things. So you see 1 and 2. So it means these two children will be having hemochromatosis. So that's going to be, so out of 4 children, it's going to be 2/4, or let's say 1/2 eventually, so to say. So that means if you're going to represent this in percentage, that's going to be 50% of the children. But since it says probability, it's going to be 2 of the children out of 4, or if you divide that out, it's going to be 1/2. I hope that makes sense. All right. Okay, so this is the next question. It says here, name the parts of the cell where you could find this. Of course, this is going to be found in the nucleus of the cell. Figure 5 shows how male and female gametes join together during sexual reproduction. So name the process. Of course, that should be fertilization. Then write down the number of chromosomes found in cell A and B, because this is humans, I presume. That's going to be 23 chromosomes, then 23 chromosomes, as it were. All right, because these are gametes, so they're going to be having half, because normal human cell has 46 chromosomes or 23 pairs, so this will be having half of that. All right. Now, protein synthesis occur in different parts of the cell. Describe the process of protein synthesis. Protein synthesis takes place by transcription and translation, but you can't just write that way because there's a way you're meant to explain that. So let's do it this way. So first point are copying of genetic information from DNA to mRNA via, or that's through transcription. Next point, the interpretation of the genetic code by tRNA via, okay, you don't have to put that, just via tRNA. All right, then assembling, okay, no, before you do assemble, okay, then when the information has been interpreted, then appropriate appropriate amino acids, excuse me, that's rough, amino acids is formed, then last part, assembling of amino acids to form protein within the ribosomes through rRNA. This is called ribosomal RNA. This is called transfer RNA. If you've seen my lecture, you've been in my classes, you understand what I mean by that. All right. So this is like a very concise way to quickly condense all the information necessary to make proteins. You have to copy the information on DNA onto mRNA. mRNA will move from the nucleus into the cytoplasm. Then in the cytoplasm, tRNA will interpret the codes on it by using a codon. Then that codon will be used to make amino acids. Then amino acids will be put together because protein is a long chain of amino acids, so to say. So that's it. So it says here, mutation during production of gametes can result in variation between individuals. Okay, figure 6 shows two complementary strands of DNA that codes for a section of protein. There's a mutation in the DNA. Circle the mutation. Okay, if you remember that every base pairs has its complementary parts. What do I mean? Adenine will always bind with thymine in DNA. Then guanine will always bind with cytosine. So A to T, T to A, C to G. Oh, this is the mutation. How do I know? Because it's meant to be G to C. So something is wrong here. So this is the answer here. All right. So these other ones are correct. I think so. Yeah. T to A, T to A to T, G to C, is to G. It's correct. Yeah. So that's the reason why we know, because it's a wrong base pair. Now, the section of protein forms part of an enzyme. Explain how mutation may affect the activity of an enzyme. Okay. So enzymes are protein in nature. All right. So which contains amino acids. Okay. Mutation will affect the genetic, let me slow down, genetic code. All right. Which in turn changes the type of amino acid formed. All right. So abnormal amino acids will alter the shape of the protein in an enzyme. Sorry, don't pardon my writing, please. In an enzyme, thereby affecting the activity of the enzyme. So what we just did here was we first established the fact that protein is formed by or forms enzymes. So the moment you affect the arrangement of the all the genetic codes on DNA, then you have affected the amino acid. Then since amino acid form protein, so you have changed the amino acid, you have changed the protein, and once you change the protein, you have changed the function of the enzymes. I hope if you can see the writing, just follow what I was saying, please. All right, that will be all for now.